Good afternoon, guys. This is Sir Franz, and I'll be giving you a review of neurophysiology before we start discussing the pharmacology of diseases of the central nervous system. So, it is important for us to understand how neurons function, that there must be a regulation of the different ions and their concentrations both inside the cell and outside cell. So first, let's have a rundown of the different important ions for the function of neurons. Sodium is the primary extracellular cation, which means it has a positive charge. The primary intracellular cation is potassium. The secondary extracellular cation is calcium. The secondary intracellular cation is magnesium, and our mnemonic for that be piso, potassium in, sodium out, and mico, magnesium in, calcium out. Piso phosphate is the primary intracellular anion, and chloride is the primary extracellular anion. Sodium and potassium have a charge of +1. That means that if their concentrations increase, they will collectively make that portion of the cell more positive. The same is seen in calcium and magnesium, but because their concentrations in the body are quite minimal compared with sodium and potassium, they do not majorly affect the voltage in a particular portion of the cell. However, their charge is positive too. Calcium will have its effects only when it is in the ionized form. Therefore, when you see blood levels of calcium, you must see the levels of ionized calcium, because only ionized calcium has a physiologic effect. Remember that calcium is sequestered in the bones of the body, and when the levels of calcium in plasma decrease, this calcium, the mineralized calcium, which has a charge of 0, is liberated from the bone and spewed into plasma as the ionized form, which is functional. So calcium inside the cell um, has a milliosmol per liter of 0, because calcium is actively regulated inside the cell. It is either pumped out the cell or pumped into certain structures of the cell, such as the endoplasmic reticulum and the sarcoplasmic muscle cells, because it is extremely potent in activating metabolism. So if you do not control the levels of calcium inside the cell, the metabolic pathways inside the cell will become extremely fast and abnormal, and that will dysregulate energy production. Calcium is also very important because it activates the apoptotic machinery. That means if you do not control the calcium inside the cell will progress with cell suicide at a much faster rate. Magnesium is the natural antagonist of calcium, and therefore, when the levels of ionized calcium are increased abnormally, these will result to neuronal hyperexcitability. That means that firing will become abnormally excessive, and this will lead to central nervous system toxicity. It will also lead to tetany or the abnormal contraction of skeletal muscles, and that is dangerous. And so, in these instances, one of the antidotes that we can give, magnesium sulfate, which will naturally antagonize calcium in its channel and in its biologic activity. Sodium and potassium are tight regulation by the kidneys. And therefore, when you have disorders of the kidneys, these will result to abnormal levels of sodium and potassium and indirectly affect function of the central nervous system. Phosphate is the primary intracellular anion, which technically just floats in the cytosol but is actively concentrated in the mitochondrial matrix. Because in the mitochondrial matrix, the phosphate anion is utilized for conjugation with adenosine diphosphate to produce adenosine triphosphate. And this process becomes dysregulated when a patient has chronic macronutrient malnutrition. That means the patient not eat the energy producing carbohydrate, fat, and protein for a long time, and you refeed that patient, the cells of the body are very hungry for phosphate because when you introduce the food stuff that will cause metabolism, production of ATP, and the phosphate be sucked out at their levels in the cytosol, and that will lead to the cells of the body sucking out also phosphate in the blood, and this will lead to acute hypophosphatemia after acute refeeding of chronically malnourished patient. This is what we call the refeed syndrome. Chloride, together with the other ions of the body, are controlled by pH. And so when there is abnormal pH in the body, so that means there can either be alkalosis or acidosis in the body. We clinicians can really look at chloride and determine what is the likely etiology or causative factor for the acid-base disturbance. Okay, next, I'd like to highlight that the fluids of the body can either be formed intracellular or extracellular, and extracellular fluid is further made up of interstitial, which is the fluid surround cells in the cells, and the other component be intravascular fluid plasma. As you will know, metabolism and function of cells will create changes in the concentration of ions and um, solutes such as glucose wastes, oxygen, carbon dioxide in the interstitial fluid. And what happens is that these fluids continuously calibrate with plasma as the plasma is circulated in the body so that there is continuous communication and recirculation. These ions, wastes, stops between the interstitial and the fluid found in the capillaries. And these fluid in the plasma are circulated by the cardiovascular system, the lungs, the liver, and the kidneys where they are processed. Therefore, there is coordination in the regulation of the concentrations of these ions and solutes. So here, I would like to introduce you to the concept the resting membrane potential. So there are a lot excitable tissues in the body. What we mean by excitable tissues are that can introduce a stimulus to those cells and those cells will respond by firing. Examples of excitable tissues are neurons, new tissue, muscles, smooth muscle, striated muscle, heart muscles, okay, and glands. So these cells are excitable, which means when we introduce a stimulus to the neuron fire. When we introduce a stimulus to muscle, the muscle will contract. When we introduce a stimulus to the glands, and the glands, and these glands will secrete. So the reason why these glands are excitable is because they are maintained at a resting membrane potential which is more negative than usual. So if you will look at this is the cell. When we say that this cell membrane is in the resting membrane potential, this means that the inside is quite more negative than the outside. And conversely, the outside is more positive than the inside. Okay? So that state of baseline negativity is what we call the resting potential. So that when we want to excite these particular cells, for example, what we do is from the baseline negative charge of the inside, we convert it to a more positive state. So that process of converting the inside to a more positive state is what we call depolarization. And depolarization is the baseline for excitability issues. So the first question is, why are these cells, particularly the excitable tissues, negative intracellularly at the side of the cell membrane? How do these cells become more negative? The primary um, effector of that state of being negative is the potassium channel. So when you say channel, it's a protein that is embedded in the cell membrane, and this protein causes facilitated diffusion, which means it will hasten up diffusion of a substance along its concentration gradient. Remember that the charge of potassium is +1, which means that if you have a lot of potassium in that region of the cell membrane, that region will become more positive. And now I'd like you to remember that potassium is present intracellularly. It is the primary intracellular cation. Therefore, the concentration gradient of potassium be from inside, which has a high concentration of potassium, to the outside, which has a low concentration of potassium. So the effect of the potassium leak channel to create a pathway for the passive diffusion of potassium from intracellular to extracellular. And as you might surmise, when potassium moves into the outside of the cell, the region where potassium came from will be made more negative. So as the potassium leak channels are active, the intracellular surface of the plasma membrane will continuously become more negative. Another effector of the negativity of the intracellular side of the cell membrane is the sodium-potassium ATPase pump. When you say that protein is a pump, means that that protein facilitates active transport. And when you say there is active transport, you will expect that the movement of molecules will against their concentration gradient, and that these movements require the hydrolysis of ATP or any other form of energy. So let us just recall our discussion a while ago and know that sodium has higher concentrations extracellularly and potassium has higher concentrations intracellularly. And if you will look at this picture, you will see that the pump functions so that three molecules of sodium or three ions of sodium are pumped extracellularly, whereas extracellularly there is more sodium. So this movement goes against the concentration. Similarly, two ions or two counts of potassium ion are pumped into the interior of the cell where there is a lot of potassium, and this movement also is an act moving against the concentration. So this is a form of active transport. But what I'd like you to realize at this point is that this movement creates an electric gradient. If you will look at it, you are pumping two cations inside and you are pumping three cations to the outside. So that means every time this pump functions, you are losing one cation to the outside. And each time this pump functions, the interior of the cell membrane continuously or negative by one count because you have lost one cation by one count. 3 minus 2 is 1. Every time this pump functions, you lose one cation from the inside and the inside becomes negative by one count. So this means that the sodium-potassium ATPase pump is an electrogenic pump. Okay. Now, a clinical application of this is that the neurons of the body will spend a major chunk of their energy expenditure just to maintain this pump. Okay, so that means that this process right here is very important for neuronal function because the neurons are spending more than half of the energy that they derive from ATP just to power this pump. And so, what happens when there is a stroke or there is a lesion, a genetic lesion that alters the production of intracellular energy in the form of when there is a stroke, there is no blood flow into these neuronal tissues. Without blood flow, there won't be delivery of oxygen that is required for oxidative phosphorylation. and production of ATP, and there won't be delivery energy substrates such as glucose, and therefore that will lead to a state of low ATP inside the neuron. And with that low ATP, this pump won't function, and sodium will continuously accumulate inside the cell. And that sodium will draw in water, and that will lead to cellular edema, and later on cellular lysis and cell death. So, with that in mind, let us apply what we have learned from the two previous slides into this final slide. So, the neuron is made up of a soma cell body and two major forms of projections: dendrites, which are afferent, and axons, which are efferent. The neurotransmitters that are present in the axonal terminal or axonal here is synthesized in the soma and just transferred along the length of the axon. And so they concentrate here, but they originate from the soma. In the soma and in the dendrites, you will see a form of sodium channel, which is ligand gated. That means that that sodium channel will open only when it receives an appropriate chemical stimulus in the form of a transmitter. So what happens is when these receptors are activated, the ligand-gated ion channel will open. And remember that the concentration of sodium is higher in the exterior of the cell. And so the concentration gradient will be from outgoing. When this ligand-gated ion channels open, sodium moves in, and that particular region of the cell membrane in the soma and in the dendrite becomes positive. And when that happens, the periphery of that ligand-gated sodium channel becomes more positive or depolarized. That will then create the stimulus for the opening of the second type of sodium channel, which is the voltage-gated sodium channel, which responds to depolarization. And as these voltage-gated sodium channels open, sodium similarly goes into the inside area of the cell. And so you have a smaller movement of sodium from exterior to interior. But if you summate all of the movement of these channels, because as I've explained a while ago, they will stimulate contiguous or near proximal sodium channels. And so the sodium channels in the periphery continuously open such that if you summate or add up the changes of charges to the movement of ions, you will create a net movement that goes from the soma to the axon, and that is the action potential. So from the soma, it travels along the length of the axon and along the length of the axon, and that is the action potential, which ultimately reaches terminal. Here in the axonal button, you encounter the third type of ion channel, which is the voltage-gated calcium channel, which responds to depolarization in the periphery by opening itself. So when it opens, the voltage-gated calcium channel will cause calcium, which is rich in the extracellular environment, to go into the inside. Once calcium has reached the interior of the cell, the axoplasm, it will cause the fusion pre-synaptic vesicles which contain neurotransmitters. The fusion will be with the cell membrane, and this fusion effectively leads to the secretion of the neurotransmitters into the synapse. When these neurotransmitters in the synapse are already numerous, they can either bind to the postsynaptic receptor or be inactivated. Okay? So first, let us look at what happens in the postsynaptic receptor. These neurotransmitters in the postsynaptic receptor, so this is the synapse, therefore this is the postsynaptic receptor. The neurotransmitters here can either activate ligand-gated ion channels, okay, or they can activate metabotropic receptors, which are G protein receptors. The activity of these two kinds of receptors differ. So when the ligand-gated ion channel is activated, it can either stimulate the next cell, the postsynaptic cell, by increasing the influx of sodium, or it can inhibit the postsynaptic cell by increasing the influx of chloride, which is negative, leading to hyperpolarization. So that positive um, influx of charges in the postsynaptic cell is associated with the binding neurotransmitters into their postsynaptic ligand-gated ion channel, and these neurotransmitters are excitatory neurotransmitters. Examples of excitatory neurotransmitters are dopamine, aspartate, glutamate, acetylcholine, and norepinephrine. However, those neurotransmitters that are inhibitory are usually coupled with the influx of chloride, inhibiting the postsynaptic cell by creating hyperpolarization. So these neurotransmitters are usually amino acid based neurotransmitters such as GABA, gamma aminobutyric acid, and lysine. Another type of receptor that can be encountered would be the metabotropic receptors, which are usually associated with the catechol neurotransmitters, the other peptide transmitters. So what happens after ligand binding into the receptor is that there is a facilitated intracellular reaction that either activates the kinases inside the postsynaptic cell, leading to phosphorylation and activation of the synaptic cell, or the inhibition of phosphorylation or the removal of phosphates in the intracellular parts of the postsynaptic cell, inhibition of the postsynaptic cell. So here in the synapse, there can be a lot of transmitters released, and so the overall effects on the postsynaptic cell is actually a summation of all the stimuli. So that is what we call the excitatory postsynaptic potential or the inhibitory postsynaptic potential. And these are summated, and the balance which predominates, whether it is the excitatory postsynaptic potential or the inhibitory postsynaptic potential, is what dictates if postsynaptic cell will fire, will not fire. Okay. So another major fate of the transmitters released into the synaptic cleft or space is inactivation. And inactivation can proceed via three pathways. First, the neurotransmitter can be taken up again by the presynaptic cleft, and the loss of transmitter in the synaptic space prevents activation of synaptic cell. Another pathway is via degradation through synaptic enzymes. So the neurotransmitters are degraded there and then their activity is lost. The neurotransmitters can basically just diffuse away from the synaptic cleft, leaving the synapse and leaving the chance to activate their postsynaptic receptors. So let us just summarize the neurotransmission process. The action potential starts off in the soma and moves down along the axon to reach the axonal terminus via the concerted action of initially the ligand-gated sodium channel and then the voltage-gated sodium channel. As depolarization reaches the axonal button, calcium channels start to open. Calcium moves inside the cell and causes fusion of the vesicles which contain neurotransmitters with the cell membrane, leading to the release of transmitters. The neurotransmitters act on the postsynaptic receptor, either activating the postsynaptic neuron or inactivating them. And then they either are taken up again via reuptake, they can be degraded, transmitters in the synaptic space, or they can basically lose, causing termination of that. So that is how neurons function in the central nervous system, technically also in the peripheral system. Later on, as we discuss the three major disorders of the CNS, dementia, or the loss of cognitive ability, mental disorders, exemplified by Parkinson's disease, and epilepsy, we will be banking toward knowledge of these physiologic concepts of the neuron and synapse and the activity of transmitters. For us to understand what is wrong with these disorders, for us to understand how current patients work. So, thank you for listening.